Welcome to Biotechnologies Allergen News 2012. I'm Sabine Wünschmann and I'm the Head of Allergen Manufacturing, Research and Development at Biotechnologies. In this video, I would like to focus on some of Biotechnologies' research highlight of 2012, which includes recent work from Dr. Anna Pomez and Dr. Martin Chapman on the structural biology of Group 1 Mite allergens and also on Alternaria. I will also talk about cat allergen Feldy-1, which has been our best-selling allergen of the year. Next, I will give a brief overview of Biotechnologies' low-tox allergens and endotoxin services, and finally talk about new allergen products that came out of R&D this year. Let's start with the research on the structure of Group 1 Mite allergens. The Group 1 Mite allergens are potent allergens that are secreted from the mite species D. theronesinus or D. farini, and high levels of these allergens are detected in mite fecal pellets. Both allergens are secreted from the mite with an N-terminal pro-region that is cleaved upon enzyme maturation. The crystal structures of DIRF1 and DIRP1 were solved through collaborative NIH-supported research studies between scientists at Biotechnologies and structural biologists at the University of Virginia. This figure shows superposition of the crystal structures of matured natural DIRF1, shown here in green, and mature recombinant DIRP1, shown in blue. DIRP1 and DIRF1 are cysteine proteases that share 89% sequence identity, so not surprisingly, IgE responses to the Group 1 Mite allergens are highly cross-reactive. Despite the high amino acid sequence identity between Group 1 allergens, more than 95% of the monoclonal antibodies raised against either DIRP1 or DIRF1 are species-specific. The DIRF1-specific monoclonal antibody 4C1 is unusual in this respect since it also strongly binds DIRP1. Monoclonal antibody 4C1 also partially inhibits IgE binding to DIRP1, suggesting that the 4C1 epitope and the human IgE epitope overlap. In a study published in JBC this year, we presented the crystal structures of natural DIRP1 and DIRF1 in complex with monoclonal antibody 4C1. This figure shows the molecular surface of natural Group 1 Mite allergens. Residues that differ in DIRP1 and DIRF1 are shown in yellow. Amino acid substitutions in different DIRP1 and DIRF1 variants are presented in dark blue, and residues forming the 4C1 binding epitope are shown in light blue. The N-terminal propeptide is shown in ribbon representation. This figure shows the structure of DIRP1 in green and complex with the 4C1 monoclonal antibody FAB fragment. The heavy chain of the monoclonal antibody is shown in blue and the light chain in purple. Resolution of the allergen and monoclonal antibody complex allowed the identification of cross-reactive epitopes between DIRP1 and DIRF1, and also identified residues involved in IgE antibody interactions. Next, we are going to move on to Feldy-1, our best-selling allergen of the year. Feldy-1 is a major allergen from cat, eliciting IgE responses in 95% of patients with allergy to cat. The Feldy-1 structure was solved by Kaiser et al. In 2007. Feldy-1 is a 35 kilodalton tetrameric glycoprotein formed by two heterodimers. Each heterodimer is composed of chain 1 and chain 2. Natural Feldy-1 is glycosylated, and the glycosylation site is indicated at residue asparagine 103. Biotechnologies' recombinant Feldy-1 construct consists of Feldy-1 chains 1 and 2 co-expressed with a linker and Pichia pastoris. The SES page on the left-hand side shows the glycosylated natural Feldy-1 at about 18 kilodaltons. And it also shows a recombinant Feldy-1 expressed in Pichia as a mixture of non-glycosylated and hyperglycosylated proteins. A deglycosylated recombinant Feldy-1 was engineered to silence the N-glycosylation site at asparagine 103 using site-directed mutagenesis. This removed the hyperglycosylated forms of recombinant Feldy-1 and results in expression of non-glycosylated Feldy-1 monomer and dimer as shown in the SDS page on the right-hand side. IgE binding of the three different Feldy-1 forms was measured using chimeric ELISA. IgE binding of both recombinant forms of Feldy-1 correlated very well to that of natural Feldy-1. An excellent correlation was also seen between Pichia-expressed recombinant FeLD1 and the deglycosylated recombinant FeLD1. Listed here are Biotechnologies' natural and recombinant FeLD1 products. The improved deglycosylated recombinant FeLD1 now replaces the hyperglycosylated recombinant FeLD1 product. Next, I would like to focus on Biotechnologies' low-tox allergens and endotoxin services. Low-tox allergens have endotoxin levels of less than 0.03 EUs per microgram protein and are designed for any studies where endotoxin may influence the cellular response. Applications include human and murine cellular studies using T cells, antigen-presenting cells, or dendritic cells, animal models of IgE responses and asthma, and in vitro diagnosis. Biotechnologies' inventory of low-tox allergens include mite, animal, pollen, and egg allergens. The low-tox mite allergens are shown on the SS page on the right. Next to the purified recombinant and natural DOPI1 and natural DOPI2 is the low-tox mite antigen DPE4. This product is specifically designed for mouse immunologists and has verified high concentrations of DOPI1 and DOPI2. Is too much endotoxin masking your results? We offer custom-designed endotoxin removal from your protein of interest. Contact us with your project and we will be happy to discuss this new service with you. Also, any allergens that are listed on our website can be ordered as low-tox allergen. This service will be of interest to researchers involved in human or murine cellular studies, animal models, or in vitro diagnosis. Biotechnologies analysis service includes testing for endotoxin. This will be of interest for occupational health monitoring, epidemiologic studies of allergic disease, and product testing. Both the recombinant factor C assay and the LIL assay are available for endotoxin assessment. Next, I will introduce two of Biotechnologies' new allergen products. Natural ragweed allergen MB1 was added to our range of pollen allergens. Ragweed or ambrosia is an important seasonal area allergen in the US, Canada, and more recently also in Europe. MB1 is a major allergen of short ragweed, eliciting IgE responses in over 80% of ragweed-sensitive patients. There are 5 known isoallergens of MB1. This page shows natural MB1 that is affinity purified from short ragweed pollen. MB1 is a 38 kilodalton pectate lyase with multiple isoforms. Natural MB1 undergoes proteolysis during purification and is cleaved into 2 chains of 25 and 16 kilodalton. Mass spec peptide mapping of affinity purified natural MB1 confirmed the identity of MB1 and the presence of different isoallergens. The mass spec data also suggests that MB101 is the most abundant protein in affinity purified natural MB1. Monoclonal antibodies that were raised against the affinity purified natural MB1 are now available for research applications. The 4 monoclonal antibodies all react with ragweed extract, but display different reactivity to MB1 isoallergens. Monoclonal antibody 5F6 reacts universally with all 5 isoallergens, while 3C7 and 5F7 predominantly react with MB1 or 103. Another new product Biotechnologies has added this year is recombinant mold allergen Alt1. Alternaria is a prevalent mold in the arid climate of Arizona. Alternaria species is one of the most common molds associated with allergic diseases. 80% of Alternaria-sensitive patients produce IgE antibodies to a major allergen, Alt1. The structure of recombinant Alt1 expressed in E. coli was just recently solved through collaborative research studies between scientists at Biotechnologies and structural biologists at the University of Virginia. The crystal structure of Alt1 reveals that it has a unique beta-barrel structure that is comprised of 11 beta strands that form a butterfly-like dimer. The right-hand side shows a surface of Alt1 with mapped electrostatic charges of the residues. Biotechnologies recently expressed recombinant Alt1 in Pichia pastoris. Under non-reducing conditions, SCS page shows the recombinant Alt-A1 expressed in Pichia or E. coli as dimers at 29 kilodalton and a monomer at 15 kilodalton. Under reducing conditions, only the recombinant Alt-A1 monomer is visible. No difference in monoclonal antibody binding was seen between the two different recombinant Alt-A1 constructs. IgE binding of recombinant Alt-A1 expressed in Pichia pastoris showed an excellent correlation to that of recombinant Alt-A1 expressed in E. coli. Applications for recombinant Alt-A1 produced in Pichia include T-cell studies, histamine release assays, and mouse models of asthma. Recombinant Alt-A1 produced in Pichia pastoris has replaced the recombinant Alt-A1 product expressed in E. coli. To summarize, we talked about several purified allergens, looked at crystal structures, and antibody binding to allergens. The use of highly purified allergens is crucial for applications in basic research, molecular diagnostics, and for allergen standards and vaccines. The advantages of pure allergens are that they represent defined proteins with confirmed IgE binding, low endotoxin content, and verified functional activity that meet defined QC criteria. Thank you for your attention. Any questions, please contact me or Ms. Lisa Vales.